I've had a really fun time putting this talk together. So I'm really hoping you enjoy hearing it. So AIDS and COVID, the thing that connects these two pandemics in Australia is me, as Sabo said. And the funny thing, not the funny thing, but the common thing in both cases is that it was actually my expertise as a medical researcher that was adopted into a public health laboratory. And this will all make sense as I go along. And so I've divided the talk into timelines. And so there's overlapping timelines, AIDS and COVID and my career. And so we start off with AIDS. And I have to acknowledge websites. A lot of this is from websites, and also I had the opportunity to speak to Bill Maskell, and so some of it is you can blame Bill for it. Anyway, so basically, as far as the AIDS timeline is concerned, in the late 1970s, 1980s, there was a rare lung infection, so PCP, pneumocystis carinii, pneumonia, which is a opportunistic infection, sort of and a cancer, a skin cancer, Kaposi sarcoma, in otherwise healthy young, gay men. So this was noticed that there was something going on. By November 1982, we had the first AIDS case diagnosed in Australia, and it was in Sydney. But by May 1983, the French group published in Science that they had isolated the AIDS virus, the AIDS retrovirus. So this is really moving ahead very fast. December 1983, there was the first death from AIDS in Australia in Melbourne, and that was at Prince Henry's Hospital. And then in April 1984, the first AIDS patient was admitted to Fairfield Hospital. And Fairfield Hospital became the epicenter for AIDS treatment and research in Victoria. Then in May 1985, Ian Gus, sitting right there, and David Pennington established a national screening of blood supplies so that the blood supply was safe from HIV. And also this test was offered on a voluntary basis for anybody who wished to be tested for HIV. And then in August 1985, Liz Neat isolates the first Australian HIV retrovirus in Australia. And I'm Liz Neat, by the way. But I will make a slight segue into Fairfield Hospital, because while I was putting this together, I was actually blown away because I didn't know a lot of this. And I have to thank Renato, sitting right there, who put me in touch with some of the information. So in the late 1800s, Victoria infectious diseases was rife, and it was decided that we needed a dedicated infectious diseases hospital. Finally, it opened as the Queen's Memorial Hospital in 1904. And all of this, progressively it built and was built upon and built upon and built upon. And it was the largest infectious diseases internationally and internationally renowned. And there was a lot of work done, particularly with polio and hepatitis viruses, particularly, with Ian Gus and his team. But this is an image of young children in an iron lung, which is just awful to think about. And this is a reporter interviewing a lady in an iron lung. I mean, things were quite dramatic in those days, to say the least. And this here is where we could actually treat patients with highly contagious, dangerous pathogens, because they were in these little isolators, and people were dressed in these sort of protective gear. Okay, so the AIDS timeline continued. So I started at Fairfield Hospital in June 1985, and I was interviewed by Ian Gus and Renato and Rob Pringle, who's not here. And I remember Ian Gus asking me on several occasions, yes, but can you grow T cells? And I remember thinking, good grief, man, can't everybody grow T cells? But of course, No, not everybody can grow T cells. We grew a lot of T cells because I was in an immunology lab at the time. And when I arrived at Fairfield Hospital, basically we had dead T cells floating around in very alkaline RPMI. But all of the protocols and all of the reagents were in place because all of the work that had been put into place beforehand. And by August, I had RT, so reverse transcriptase in the supernatant confirming that we had a retrovirus. But this sort of graph is a bar graph or a line graph, so it's pretty boring. And Ian Gus really wanted an image. That's what sells the field to politicians. So John Marshall was strapped to the electron microscope until he got that image. And that Thursday morning, when John Marshall came down the corridor saying that he had that image, was an absolute eureka moment for the entire hospital. And it's one of the highlights of certainly of my career. And I also got to work with fantastic infectious diseases physicians. And I just want to point out Anne Mitch in particular because I got to know her quite well because later in 1986, we went to the International AIDS Conference. And, and and we were chatting and Anne was saying she decided, to specialize in infectious diseases because, people come in, you treat them, they go home. It's all nice. It's a nice thing to do as a doctor, as a clinician. And then along came AIDS. And so she was just, I think she went to, every funeral and did so much work closely, related, closely associated with the patients. But at this conference, we shared a room, and this is just to typify what sort of dedication Anne had. She had a patient who was on some sort of a respirator, machine, whose dream it was to go to Paris. And Anne took the patient with her. So the three of us shared this room in Paris, and Anne took care of her, which is just, amazing. And then later that year, I went to the WHO meeting again in Paris, and this was to standardize and procedures and protocols for isolation. Then I took maternity leave from the AIDS isolation job and came back in part-time in various different roles. And one of them was to establish the flow cytometry unit once the centre that was established by Ian, the McFarlane Burnett Centre, attracted funding to buy a FactStar Plus. Now, this was the most U-Butte instrument in flow cytometry. In those days, it took up an entire room. Today, it's the size of a photocopier. But we established, a state-of-the-art protocols, and one of them was a whole blood lysis assay for enumerating CD4 and CD8 T cells. Now, that is a very important diagnostic result for HIV because HIV kills the CD4 positive T cells. So if the numbers get down, it's becoming critical. And then I decided to do a PhD with Dale McPhee, and I was co-supervised by Bill Boyle. And then by the end of my PhD, I left to write up at home and then left Fairfield. And then in the 1990s, Joe Manita, who was already at Fairfield, adopted the whole blood lysis assay into the Fairfield Hospital Clinical Pathology Services. So there was this fantastic interaction between clinicians, infectious diseases, physicians, and medical scientists, and it was just a wonderful place to work. And the reason that we could isolate HIV so quickly was because there was a lot of infrastructure, an enormous amount of medical scientists and infectious diseases talent already at Fairfield. And I've already mentioned I forgot to mention John Marshall. But anyway, I think I did. John Marshall for electron microscopy. There's Liz Neat with her T cells. And Rob Pringle, who had a Midas touch for establishing serological assays. Joe Manita, I've already mentioned. And there was a couple of laboratories, like a general virology culture lab, viral ID lab, and also specialized laboratories. And in those days, Cells were grown on glass little files, and people here are looking for cytopathic effect to identify, to show that there's virus there. But all of this was under the inspired leadership of Ian Gust, who was the director of virology at Fairfield, 1970 to 1990, and established the McFarlane Burnet Centre in 1985. And so the flow cytometry unit was within the centre. And a lot of the infrastructure, as I said, was already established at Fairfield. So we already had the high security lab. It was already built. A lot of work must have gone into designing it and making all the decisions that go with it. But because this was a high profile, disease, HIV, and we had a lot of politicians coming in to see what it is that we were doing and to see what has happened at Fairfield with the money that has been invested. So we were tasked with doing all the HIV isolation work in the high security lab and had to work in Pathoflex class 3 cabinets. So we're working through gloves, several pairs of gloves, and I think we grew more fungus than HIV, but never mind. And then also I was, my first RA was Jeremy Jowett, who's about 6'6", and me, I'm about 5 nothing. And so I was, and we wore these overalls. So we went through a shower airlock and then, changed into these overalls. And I was tasked with choosing the colour for the overalls. And because the entire lab was very pale and pale blue and whatever, and I thought, if we faint, I mean, we're out there isolated. People knew we were going out there, but we were out there for hours doing tissue culture. So I thought, if we faint, no one's going to see us if they happen to be walking past. So I went for blood red. But we managed to puddle our way through and I reread this paper, and I think we did some remarkable work, to be honest, and published the Australian experience with HIV isolation. And we actually stayed at the end, near the end of the paper, that we doubled our success rate of isolating HIV from T cells once we switched to using a class 2 tissue culture cabinet. So it's much easier to work in a class 2 cabinet like this. So now I'm just going to, again, diverge for a second and talk about the bitter dispute, which at the time I wasn't completely aware of this, between Luc Montagnier and Robert Gallo. And this went on for 10 years. And the information that I'm presenting is from this article in the British Medical Journal. And so in May 1983, Luc Montagnier's team at the Pasteur Institute successfully isolated a novel virus from an AIDS patient, which they named LAV. And then Luc Montagnier sent his virus to Gallo for confirmation. Robert Gallo was, and I think still is, the leader in retroviral virology. And there was a written understanding that it would not be used for commercial gain. So April 1984, Robert Gallo's team in the US announced that they've isolated the virus that causes AIDS, and they called it HTLV3. So Bob Gallo had isolated HTLV1 and also HTLV2. So it made sense for him to call it HTLV3. But then in March 85, Rob Gallo's team developed the first widely used blood screen test. It was FDA licensed. But during that year, molecular biologists were sequencing the entire genome of AIDS, HIV isolates, and they showed that the genetic sequence of LAV and HTLV3 are identical. So they could smell a rat. So December 1985, the Pasteur Institute filed a suit against the US government, and this, went on for 10 years. Now, I was unaware of this sort of at the time, and I happened to trundle along, I think it must have been middle of 1986, because I'd gone overseas to learn more about HIV, so that we're, up to date at Fairfield. So I spent, I think it was a couple of weeks in Bethesda at NIH, including Bob Gallo's lab, then a couple of weeks at CDC in Atlanta, Georgia, and then went to the Pasteur for a couple of weeks. And I was tasked with bringing back to Australia the French, virus, and I think it must have been LAV2. But nonetheless, once I was, this is a very familiar photo, scenario. We were in the lab and yakety yak, working out how we're going to, package it and all this sort of stuff. And then Bob Gallo discovers that I had just been at NIH Luke Montagna discovers that I had just been to Bob Gallo's lab at NIH. And then there's an explosion of French that thankfully I did not understand. And Professor Sherman, who's a lovely man, scuttled me away from all of this hysteria and says to me, "Oh, you and I must have a rendezvous." And I thought, "Oh, okay." I hope that means a meeting. Anyway, which of course it did. And so he apologized profusely for Luc Montagne's behaviour, and we cooked up a plan of how I was going to courier the virus into Australia, and they would take care of the paperwork later. But less said about that, the better. Anyway, so in the end, Robert Gallo was cleared of research and misconduct. Some people felt that the end justified the means because the test that he established saved lots of lives, which is true, it did. But on the flip side, if people had have taken Luc Montagne's work seriously, there would have been an assay a year earlier. Both papers were published in Science, so it wasn't like one was buried, somewhere, but people ignored this first publication. But anyway, in the end, it was actually Françoise Barré-Senoussi and Luc Montagna who were awarded the Nobel Prize in 2008 for their discovery of the human immunodeficiency virus. And to be honest, I actually agree with that finding, or that decision. So part two, so this is Wnt signaling cancer and organoids. And the reason I'm going into this part is so that we understand why it is that the work that we did during COVID, why we did the work during COVID and how it all sort of happened. So to explain what organoids are, they're simply growing tissue replicas in a dish. So organoids are three-dimensional, they're stem cell derived, and the stem cells self-organize into organoids. And it revolutionized science and medicine because they bridged the gap between simple tissue culture and then in vivo mouse models. And if we do a search in PubMed for the term organoid and look at the number of publications, there's an exponential increase after about 2009. And the reason for that is the first paper from the Cleaver's lab on tissue stem cell derived organoids was published in 2009. So what I'm getting at, broadly speaking, there's two approaches to growing organoids. And so the first one is pluripotent stem cell derived organoids. And this is where cells are taken back to a very primitive state and then taken through all of these different developmental cues and differentiation cues. And we end up with organ-like structures at the end. How well this works depends on how well these cues are known. And irrespective of what anybody tells you, these end products are often still fetal, slightly fetal. The second variety is tissue stem cell generated organoids. Now, tissue stem cells remember their tissue of origin. So if you take an intestinal stem cell and put it somewhere else, it'll still make intestinal epithelium. So it knows what to do. So they're already different to pluripotent stem cells. They generate the cells they normally generate, and consequently, they recapitulate tissue characteristics and function. And so we grew our first organoids in 2010 and have grown organoids from a whole lot of different tissues including cancers that arise in those tissues. But just to drive it home, because people use the word organoid loosely, it actually means stem cell derived. So you start off with one cell, and this is from Toshi Sato's paper in 2009. And so this single cell is taken through and eventually forms this lovely three-dimensional structure, an organoid. And the LGR5 is coupled to a GFP reporter. So you can see green cells at the tips of the crypts, which is where the stem cells are. So this is a little cartoon to sort of build on that. So this is the epithelium with the villus domain and a crypt domain, and I'm using the color coding from their papers. But I imported the mice back in 2008, 2009, and when we managed to get funding. And so the way that you establish organoids is we chop off the crypts and put them into matrix, supply all the goodies that the cells need, and we end up with organoids. And so this is a fresh crypt, just looking down the microscope, and you can see the stem cells. So that we could just, this is the first time we could look down a microscope and see the stem cells. And within one day, they start to organize themselves. And within 3 to 4 or several days, they form these organoids that have domains that are reminiscent of the original structure. So they have a crypt domain and a villus domain. And we know from the work that the Cleaver's lab did that Wnt signaling is essential for generating organoids. And so I'm just going to sum up 20 years of research in one click of the mouse. So what we showed was that Frizzled7 is the receptor that receives that Wnt signal, and that the necessity for Frizzled7 is hijacked by cancer cells. So that property is hijacked by colorectal cancer cells. And that's another whole seminar, so I won't go into that. But the work that we did was recognised by the Wnt field internationally. So this is all of us at the Wnt meeting in 2018. And other people use the knowledge gain that we generated to make therapeutics that target Frizzled7, either for cancer or for regenerative medicine. And the Frizzled7 project has now been taken forward by Toby Fessy, who was in my lab, but now he's his own lab head in Cardiff University, and he's collaborating with the people that I used to collaborate with on this project. And so is Juan now. But we were internationally recognised. I'm the only Australian ever invited as a speaker to the Wnt meetings, and I know Bang's going to say, stop talking about Wnt signalling, but I think it's important. And, eventually they learned how to spell my name. And so this is just to acknowledge the Wnt cancer organoid, collaborators and people that we work with, Hans Cleavers in particular, Nick Barker. We did a lot of work initially in EMT. So I don't know if there's any EMT people out there, but with Thomas Brablitz, he's a big shot in that field. And this is the lab in 2015. So it's the core of the lab, 3 PhD students and Toby Fessy as the co-lab head, as I mentioned. And these are the people that were in the lab for an extended time, RAs, PhD students, then stayed on as postdocs. And in 2015, we had a mature-aged honours student with very dubious credentials. I mean, that, of course, is Hans Cleavers, who did a sabbatical with me, co-hosted by Doug Hilton at WeHi. And you might wonder, now, why on earth is this guy do his first sabbatical in Australia? And that's because I kept trying to, it was really hard to get funding for Wnt projects. It was and probably still is an underappreciated field in Australia. So I thought I'll bring the Wnt field to Australia and organised an EMBO workshop, which I made it in Cable Beach, Broome, which was absolutely idyllic. And everybody loved it. but we had 130 delegates and most of them were international. So Australia didn't really turn up, but we had a marvellous time. And that led to him wanting to come back to do a sabbatical in Australia. And this is my final slide on wind signalling. As a straight medical researcher, I'm not a medical person. We do a lot of medical research, but I'm just a straight researcher. And I think the wind signalling pathway is the classic pathway for bench to bedside. Now, who would have thought that experiments in the frog or a freshwater polyp or indeed any of these other developmental and lower organisms would actually end up being something that is now being exploited for therapeutics. And you heard a lovely talk recently from Ricky Johnson about personalised medicine and being able to grow cancers. So on to enough about COVID, enough about AIDS, enough about wind. We're on to part 3, which is the COVID timeline. And I really like this picture, but it actually sums up the very early stage of COVID in Australia. Julian Druce was the head of Viral ID. They were the first people outside China to grow SARS-CoV-2. So that was in January 2020. And the Doherty is a joint venture between the University of Melbourne and the Royal Melbourne Hospital. And this photo typifies that. So Christine Kilpatrick, who was the CEO of RMH, Julian, as I said, was the head of VI and isolated SARS-CoV-2. Professor Kshitij Kapoor was the Dean of FMTHS, University of Melbourne. And of course, we have Mike Catton, who was the director of Vidril at that time. It was a very exciting time. But here's the COVID timeline to put that into context and just how fast things happened in Australia. So day one, and this is slides that I've adapted from a slide deck that Julian Druce and Leon Carley shared with me kindly. So day one, the first case is identified in Wuhan, China. It took them about 3 weeks to notify officially the WHO, and that market was all of the cases were linked to a market in Wuhan, and the market's closed down. But very early on, January the 11th, Edward Holmes from the University of Sydney, he was collaborating with the Chinese researchers who were sequencing the SARS-CoV-2 genome, collaborating with them to get the full sequence. Now, he convinced his Chinese collaborators to publish the sequence, even though they had a gag order to not share data by the Chinese government. So very brave and very important, because once you have the genetic code, then you can design primers so that you can specifically identify SARS-CoV-2, which is exactly what Leon Carley did at Vidril. Within a week, I think it is, they had their first PCR positive sample. So, it was... I remember the day actually. Anyway, they had their first PCR positive sample. Because Julian had already been alerted by WHO that there's something going on in Wuhan, they were already constantly having tissue culture cells ready to go. So if they were managed to get a PCR positive sample, they could put it into culture straight away, which is exactly what they did with this specimen. And then again, in no time at all, Jason Roberts, who was now doing the electron microscopy at Vidril, had the image of SARS-CoV-2. So all of this just went boom, 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 really quickly. And then there was this huge media, it was just a media fest. And they were able to publish the work fairly quickly. It was Julian's insistence that we share the virus because it's a public health laboratory. You share the information that you have, you share the viruses because we're responding to a pandemic that kills people. And there was a lot of media attention. And it was really, in the end, I think they sent VICO-1 is the first isolate. They sent it to over 40 laboratories, and more than 30 of those laboratories were overseas. Often you look at, phylogenetic trees and VICO-1 will be there. And all of this, again, was under the inspired leadership of Mike Catton, who was the director of VIDRAW at the time. VI isolating SARS-CoV-2 put the Doherty on the map for COVID. And everybody at the Doherty pivoted whatever expertise they had to COVID. And for my lab, it was organoids, as I've already told you about. That's what the expertise in our lab that was applicable to COVID. So back to the timeline before we get to our stuff. So January 30, WHO declares COVID a public emergency. So things are, hotting up. And we were no lockdowns yet. So I went to a meeting at CSIRO, so the Australian Centre for Disease Preparedness, and it was ex vivo models, so organoid models of all sorts of different varieties in Geelong. And I met Shafag Waters, who happens to be working on nasal basal cells and nasal organoids because she's a cystic fibrosis person. And I thought, wow, that's something that we can do. And so March, WHO declares the outbreak of pandemic. And then Jo Teresi and I established a collaboration with Shafag Waters so that she could send her frozen basal cells to us, and we could, following her protocols, set up nasal epithelial organoids. Now, because we were working on COVID, we could keep her lab open, and we also kept our labs open. But this was happening overseas as well. Laboratories were shutting down. All they could do was COVID. And within 6 weeks, a collaboration between Bert Haagmans and Hans Clevers, they were able to show that SARS-CoV-2 infects organoids. So this was the very first paper, May the 1st, online. May 15, 2020, Lim-Fa Wang already developed the first commercial kit to detect neutralizing antibodies. It was FDA approved and launched. So things were, moving really, really quickly. These moved quickly because they had their collaborative, agreements and the ethics all in place. It took us a little bit longer. So it took us until June to finally get a collaborative agreement between the two universities in Australia. And then we received our first shipment of basal cells. And by October, we'd infected the human nasal epithelium with VICO-1, the very first isolate. And we published that sort of very early in 2022. So it was online 13th of January. So what we did here, and why this is important, is that Shafag would send us the basal cells. These are little inserts that you grow, and basal cells are the stem cells of the nasal epithelium. And we lift off the medium, and once this is actually confluent and formed a tight epithelium, and let them differentiate at air liquid interface. And so it makes an epithelium that's reminiscent of the nose. And so what this actually looks like, basically in cross-section, there's the bottom, the nuclei, cilia are at the top, and there's lots of nuclei all the way through. So it is a pseudo-stratified epithelium. And we showed that it was highly susceptible to SARS-CoV-2 infection. So this is cilia at the top, which are labeled green, SARS-CoV-2 and nuclei. But what we wanted to, soon it was recognized, sorry, I forgot this bit. Soon it was recognized that SARS-CoV-2 is not just a respiratory infection. It was actually described as a rampage of the virus through the body. So globally, organoids from diverse organs were adopted to understand the pathogenesis and the target cells of this virus. Now, if we go back to our PubMed search again, we see that exponential increase as diverse fields adopted organoids, be it cancer, be it stem cells, disease modelling, except for infectious diseases, people. They were still hanging on to their simple continuous cell lines. And it wasn't until COVID that it sort of started to take off. And basically COVID put organoids on the map for infectious diseases. Now, while we were doing this, we wrote a review to summarise, because I always feel like if you're finding something, you need to actually publish it so that it's shared as quickly as possible and other people can do the same. And so I'm just going to summarise two main findings that we talk about in the review. And one of them is that organoids model authentic infection. So what actually happens with vero cells, viral entry is different to entry of the virus into, our epithelium and into organoids. And drugs that showed efficacy, like hydroxychloroquine, in vero cells, failed clinical trial, and there was a lot of money wasted on trialing drugs that were never going to work in a human. And so if they had tested hydroxychloroquine in organoids to begin with, it never would have gone into clinical trial. Also, vero cells have a defective interferon response, which is the cell's natural response to prevent infection, which is great if you're trying to just, isolate the virus. But if you're wanting to do drug screens or try and work out pathogenesis, it doesn't make sense to do it in that sort of a cell. So that's the first thing. The second thing was continued propagation of SARS-CoV-2 in vero cells eventually ended up selecting for viruses that have, subclones that have deletions in the spike that affect pathogenicity, transmission, and also drug antibody sensitivity. So that means that, why would you bother doing a drug screen in these cells? But anyway, people still did. But there is another cell line, CALU3, which is an adenocarcinoma cell line, a human cell line that was reported to preserve spike authenticity. And so we and others adopted CALU3s to grow our viral stocks. But there were other caveats and issues with CALU3s that I don't have time to go into now. But the impetus for us setting up a nasal epithelium organoid-based neutralization assay was because our hypothesis was that if you're using a cell that most closely mimics the real tissue, you're more likely to be able to get data that is directly translatable to humans. And that is exactly what we did. So this is showing a vaccine that was a VLP vaccine developed by Joe Torresi's group. And we tested the sera from vaccinated mice on the nasal epithelium to see whether the serum would prevent infection. And sure enough, in our nose assay, Joe's vaccine prevented, the sera from the vaccinated mice prevented infection to the level, basal, as our control neutralizing antibody. Similarly, this was matched by preventing infection in a mouse model that Mark Pellegrini had at WeHi. So Joe's vaccine-induced sera prevented infection in noses and also in mice, but not necessarily in simple cell lines or surrogate neutralization assays. Now, at the time, we didn't understand this. We thought, we've done something wrong. What is going on here? And recently, others have now actually shown us exactly what's going on here. And I can add a third take-home message is that organoids recapitulate and predict real-world efficacy of monoclonal antibodies. Now, what happened here is that a monoclonal antibody that was developed as a therapeutic early on in the pandemic, was used in the clinic, worked perfectly fine. Then as new variants emerged, particularly the Omicrons, the more recent Omicrons, it was shown that in simple cell lines, that neutralizing antibody no longer prevents infection. So the US withdrew that monoclonal antibody from the clinic, but Europe and the UK did not. And so what they went on to show was that the data from the Europe and UK showed that that monoclonal antibody actually worked better than the antivirals that they were using. Now, why is this happening? And the easiest way to explain this is in this little diagram. So this is SARS-CoV-2, and so this is the spike. And there's it's actually made up of the S1 domain and the S2 domain. So, very creative naming there, spike, S1, S2. And so there's class 1 and class 2 monoclonal antibodies. They work here. And that's what most of the vaccines are. They're actually just either the very tip of it or the RBD or the S1 domain, all of the vaccines that we've had. But Joe's is a VLP vaccine, so it's a viral-like particle. And as the name suggests, it's the full spike protein. And it turns out that class 3 monoclonal antibodies that work at this bit also neutralize. So now we're, frantically trying, before we both expire with our careers, trying to attract funding to develop Joe's vaccine into a platform, not just for SARS-CoV-2, but into a platform for respiratory viruses. And we're trying to attract funding to really establish the nose-based assay into something that we can, offer on a fee for service. But, he's hoping. So lots of people to acknowledge. A whole lot of people came together during COVID, mostly from DMI. And so these are all the people. It was a really incredible time scientifically. And we were lucky to get some, funding. And I actually ended up using my NHMRC Ideas Grant. We pivoted it to COVID by keeping the themes of the grant the same, but obviously not what we were planning to do. So anyway, NH and MRC were fine with it. We couldn't do anything. We couldn't do the work that we had intended. So it made sense to use the work for related work. And I particularly want to thank Joe. He and I have been working together for quite some time on organoid-based models, lots of work in hepatitis. And Bang, who's either established the organoids that I've talked about or been involved in the studies or train people to do it. And we celebrate each publication with a champagne, which I highly recommend. So it's like I was doing with Wint. I wanted to champion organoids. So I started doing these symposia in 2018 called Organoids Are Us, because organoids, from tissues really are us. And so the first few were in the Parkville precinct, and now we've taken it interstate and overseas. Highly successful symposia, heaps of people. And unbeknown to me, Yong Wen-yu had also started an organoid series of symposia in Seoul in Bangkok. Not in Bangkok, Seoul, South Korea. And he also started in 2018, but their meeting now is huge, like thousands of people. We keep ours nice and small and intimate. But I was invited to speak at the Organoid Developers Conference last December in Bangkok. Just an amazing meeting. And they're really pushing forward with organoids. They describe it as the next transformative event in medical research or science, like working out the DNA. So, I sort of think so too. But anyway, others may not. But now we've we collaborate. And so we're going to have Organoids Are Us sessions at ODC and ODC sessions at Organoids Are Us. So really broadening our reach. And I just want to, I don't know how I'm going for time. I think I'm going okay. Fine. So I just wanted to just say a huge thank you to, people who you don't realise until you get to my stage, just the impact that they had on your career. And so the first one is Bill Boyle. As Davo mentioned, I did my honours with Bill Boyle. So he's the one who taught me how to grow T cells. And then I eventually left, but in 1991, when I did my PhD, he was again my University of Melbourne supervisor, and I did my PhD through DMI, but I had a Fairfield virology supervisor, Dale McPhee, who was a lovely bloke. But I've kept in touch with the Boyle lab, and we still get together every now and then. It was just a wonderful lab to work with, and DMI is still a really collegial, lovely department. The other person is Ian Gust. Thankfully, he gave me a real job in 1985. I was sort of puddling along here in immunology, and thankfully, Ian didn't need a clever immunologist. He just needed somebody who knows how to grow T cells. I certainly was not a clever immunologist. But in those very short time, I think 5 years, we set up the first HIV isolation lab. I took maternity leave, came back part-time and did, smaller jobs. We did the first NADA accreditation, I believe, I think in Victoria or maybe Australia. I know Renato can correct it. And then I, ran the flow cytometry unit and established some novel, very innovative assays. So it was a really wonderful time. The third phase that is very important for a medical researcher is the transition from being a senior postdoc to being a lab head. And I have to acknowledge Rob Ramsey for that. We were photobombed here by some steam rail Victoria enthusiast, but never mind. It was Rob who actually helped me exit Peter McCallum so that I was equipped to set up my own lab at the University of Melbourne. And I'm still a lab head at the University of Melbourne. And finally, obviously, Nick Barker and Hans Cleavers. I first met Hans in 2002, or met him properly in 2002. Nick, I met properly in 2005 and convinced him that he should make a Frizzle 7 Flox mouse for me, which they did. And we've had several grants together and several papers. And it's just been an incredible collaboration. Hans and Nick accept all of my invitations for whatever meeting I'm organising. And then just a little note about medical research. This is what medical research usually feels like. You're hanging on, if you're a small lab like mine, small university lab, you're hanging on for dear life. And I mean, my lab was on the brink of extinction several times, and we've managed to somehow or other survive. But one of the reasons we did survive is I didn't have to worry about my salary because I had two hats. I had a regular diagnostic job as well as running a research lab. So I didn't have to focus on getting funding for my salary. I just had to focus on getting funding for staff and, students and support. So that actually takes a real load off, which was a blessing. And I have to thank my family. I am a product of, obviously your family. So my mum and dad, my brother, I love this photo of Alex and me. I mean, we didn't want to get our photo taken, but there's no saying no to my mum. But look at the faces. My partner in crime for almost 30 years, and my beautiful kids. And this photo was actually taken by Julian Drews, who's a fantastic photographer. So coming to the end, AIDS and COVID, A Tale of Two Pandemics. Obviously, it's from the book by Charles Dickens. And so it was the best of times. It most certainly was the best of times. During COVID, they talked about unprecedented level of collaboration. Oh, what a load of nonsense. There was enormous collaboration during the AIDS years as well. Fairfield was really well connected internationally recognised. The speed, we did have phones in those days, so it wasn't that much slower than it is today. But what the difference is the technology. The technological advances from the mid-1980s to now is just mind-blowing. And that's why so many things could be developed so quickly. It was the worst of times, most certainly. Lots of people died. There was a lot of fear and anxiety within the community, and I think some of us still get PTSD from some of the, if you see a photo. It was the age of wisdom, most certainly, and I was lucky enough to have really inspirational and hugely talented, well-connected leaders and bosses. It was the age of foolishness. And I thought the word foolishness seems really weird. I mean, what does it actually mean? So I Googled it. And what it means is that this is what it means, exactly from the internet. Behavior, speech, or decision that lack good sense, wisdom, or sound judgment. Now, unfortunately, during both pandemics, near the end of my time there, both were true. But less said about that, the better. So thank you so much for listening, and thank you. I'm an Alice President fan, so thank you very much. And I'm just going to put up this slide. This is all the lab members, past and present, collaborations or collaborators that we did a whole lot of work with that weren't part of AIDS or COVID. And I'm happy to take questions.